Welcome to the official podcast of the Ogden Surgical Medical Society. Since 1946, the Ogden Surgical Medical Society has been educating our medical community. In this podcast, we bring you the voices of medical experts in your community and around the world. Please note that the opinions and ideas shared on this podcast are of the speakers, and not of the opinions of the Ogden Surgical Medical Society or its board. All medical topics discussed are not to be taken as strict medical advice, and individual clinical judgment is warranted when applying information to patient care. It is the responsibility of the listener to judge for themselves the applicability of the information discussed in regards to their medical practice.
Today, we are so excited to have Dr. Martin Tristiani Firouzi today speaking with us. He is from the University of Utah. He's a professor of pediatric cardiology, and he's an expert in precision and genomic medicine. My name is Dr. Scott Moore. I am the current president of the 2023 Ogden Surgical Medical Society, and I am a professor of medical laboratory sciences at Weber State University. I teach clinical chemistry. And with me today, I have Heather Hyleson. Heather, you want to introduce yourself?
Sure. Thanks, Scott. I'm Heather Hyleson. I'm an emergency physician with Utah Emergency Physicians, and I am current co-chair of the Ogden Surgical Medical Society.
Dr. Tristiani, he earned his undergraduate degree in St. John's University in Natural Sciences and Chemistry, and his medical degree, pediatric residency, and pediatric cardiology fellowship, that's a mouthful, at the University of Minnesota. Dr. Tristiani's research is dedicated to the study of and practical application of precision medicine. Really, really cool stuff. His lab has built an infrastructure supporting precision medicine by connecting disciplines that excel in genomic analysis, functional analyses, and healthcare information technology. Now, Dr. Tristiani currently serves as the co-director of the Center for Genomic Medicine at University of Utah's premier Center for Genomic Innovation, Discovery, and Translation of Genomic Medicine into improved patient care, which is really what we care about. This is the coolest part. In this role, he supports efforts to create unique analytic tools, collaborative teams, and clinical pipelines to diagnose critically ill children with complex phenotypes and to end diagnostic odysseys for children with undiagnosed genetic disease. Now, the Center for Genomic Medicine actively supports endeavors to make precision medicine, including gene therapies, a reality at the point of care. This is groundbreaking stuff. Now, in his spare time, Dr. Tristiani enjoys reading about ancient DNA from our human cousins, the Neanderthals, and I'm going to butcher this because it's not like everyday terminology, the Denisovans? Denisovans, okay.
Denisovans.
Now, as well as he also does early human migrations out of Africa and the advent of agriculture in the setting of first human civilizations. And that's something that's cool, but we are so glad to be able to have an expert like you in this. And you know, Dr. Tristiani, if you don't mind, I like to be on a first name basis. I'm Scott. I'll call you Marty. That works.
That sounds great.
Perfect.
You can call me Heather.
Perfect. So, you know, I'm just wondering, you know, your bio says so much, but then how do you correlate all of that with some of this old ancient information that we're getting from the Neanderthals and the Denisovans?
Yeah, so it sort of fits in with my interest in genomics and genomic medicine, right? And so you know, the Nobel Prize was given to a German investigator who was responsible for sequencing the Neanderthal genome. And that was a few years ago. And that has really allowed researchers to understand, first of all, that humans, Homo sapiens and Neanderthals mated and had viable offspring that were able to have children in and of themselves, which was a shocking result at the time. Most situations where you have unique species are unable to mate. They mate, but then the offspring are infertile. And in this case, here's a situation where there was mating between Homo sapiens, us, the humans, and Neanderthals, our cousins, that resulted in viable individuals. And so that sort of, the two ideas sort of feed in together within that concept about using DNA to understand in my research to understand genetic problems and therapies. But then in my spare time, just kind of fun things is to understand human history and how we got to where we are, and human evolution, and then just the whole idea of how people came together to cooperate to build the first cities, to have a hierarchy that allowed different forms of work to be distributed across different areas, which then, of course, resulted in art and pottery and all these other, and writing ultimately, and then our modern technologies and civilization.
It just sounds amazing to think of how far this goes back and how we can track that. It really gives us kind of a glimpse into history that we've never really had before. That's
exactly right.
So Marty, we've heard a lot in the medical field about this new technology called CRISPR. And I'm assuming that it's not referring to those, crispy, like rice puff things inside of candy, which that would have worked really well for my conference. But for this conference, think about how this is, this CRISPR technology can have its place in medicine and how it relates to, Nobel Prizes in medicine. And, what exactly is CRISPR, the 10,000-foot view?
Yes. Yeah. So the original ideas of this research date back to the late 1980s, but really our understanding of CRISPR takes off in the year 2000 and onward. So CRISPR is a mechanism used by bacteria to protect against invading viruses or bacteriophages. So these bacteria produce pieces of RNA that are complementary to the viral genome, and this is linked together with an enzyme called an endonuclease that actually cleaves the DNA and thus inactivates the invading virus. So these early pioneers in the research field understood the potential of harnessing this bacterial system to specifically edit the human genome, work that ultimately led to the Nobel Prize in Medicine in the year 2020. These individuals are Drs. Emmanuelle Charpentier and Jennifer Doudna. So the CRISPR system, sometimes it's called CRISPR-Cas9, or sometimes just CRISPR, consists of a guide RNA that's linked to the Cas9 endonuclease and includes a specific DNA sequence called the PAM sequence. So let's break down those individual pieces of the CRISPR machinery. So the guide RNA includes the complementary sequence to the DNA site that you want to target. And by target, we mean the DNA you want to cut. This PAM sequence is a crucial anchoring point for the DNA. So the CRISPR complex actually anchors to the PAM sequence, and the PAM sequence is just an existing part of the DNA structure. All right, and then the other component is this Cas9. And the Cas9, this endonuclease, is an enzyme that cuts both strands of DNA at the target sequence. So this idea of harnessing this bacterial machinery in order to specifically cut DNA at your target site with very limited off-target cutting was the reason for the Nobel Prize designation.
Sounds to me like CRISPR, are we talking about like primers here? Kind of like when we have like DNA replication, are we talking like primers?
Sure, that's exactly the way you can think of it. So a primer is a series, usually about 20 nucleotides, which is complementary to the DNA that binds, and then you do something with that, whether you're doing, say, PCR, where you're amplifying those regions. And the guide RNA has that complementary sequence. And the reason why CRISPR is so efficient and specific is that when you look at across a 20 nucleotide sequence, the chances that you're going to have that identical sequence elsewhere in the genome is relatively low, with few exceptions. But it's sort of then oligonucleotide, or that RNA guide is actually linked directly to the Cas9 endonuclease and that PAM sequence identifier. And it basically scans through the entire, DNA sequence until it finds the match, and then boom, it locks on there like a key to the, to the complementary sequence, and it requires this PAM recognition site that anchors it, and then the enzyme is sitting right there, and it snips the DNA and does a double-stranded break.
I love enzymes; those are so cool.
That's so interesting. Now that we understand how CRISPR technology works with a pan sequence, kind of identifying that, locking on, and the kind of the endonuclease. And it's so cool that they got the Nobel Prize for that in 2020. I did not know that. What are some applications for CRISPR technology in medical research in general, or your medical research that you're involved in?
Sure. So we use CRISPR a lot in my lab to knock out genes and then define the function of the gene by eliminating its expression. So you may remember that back in 2007, Dr. Mario Capecchi from the University of Utah received the Nobel Prize in Medicine for designing the technology to knock out genes in the mouse, which then paved way for our modern understanding of gene expression. So CRISPR markedly simplifies this ability to specifically knock out genes by virtue of its high specificity of this guide RNA binding to the target site with minimal off-target binding. And so a few years back, we published methodologies for knocking out genes in the zebrafish using CRISPR technology, which turns out to be fairly straightforward. Our goal at that time was to understand the function of genes in which we had discovered mutations in children with congenital heart disease. And we wanted to characterize the function of those genes in the developing heart. So that was a model organism where we would introduce, basically knock the gene out and follow heart development in the zebrafish and say, hey, this gene is important, had never been associated with congenital heart disease in the past, but now we've created a link because we have patients that have a mutation. We've knocked the gene out in the zebrafish, it alters cardiac function, and so create sort of a link between human disease and a new gene that is important for congenital heart disease. So this is kind of like the simplest use case for CRISPR, and that is to create a double-stranded break. So what happens when the DNA is cut by the endonuclease is the cell's internal repair machinery tries to correct this break. And when correcting it, it very often adds or deletes a base pair here, a few base pairs here and there, resulting in what we call an insertion or a deletion. We call that an indel, and that shifts the reading frame of the DNA, resulting in a truncated gene transcript and then a non-functional protein. So if you include a piece of DNA that's homologous to the target site, but includes a point mutation, then that cell DNA repair machinery might swap that donor DNA right at the target site, resulting in the introduction of a point mutation. And that's something that's called gene editing or genome editing. The problem with this genome editing approach is that this introduction of the donor DNA is a very low frequency event. So you have to screen through thousands and thousands of clones to find the correct event. And so that's really been a major limitation in sort of the gene editing technology. But compared to the tools we had in the past, this is a step up because of its high specificity. So in thinking about the application of research, or thinking about the application of CRISPR for research, that includes the ability to specifically, efficiently, and rapidly knock out genes in a cell line or model organism in order to study the gene function. And then in addition, you can knock in specific mutations, like a patient's mutation, into that cell or that model organism and study the impact of that mutation. And so this knock-in portion of the gene editing is, of course, very laborious.
Yeah, I mean so now knock in. I've always assumed that you would take out bad mutations, or you would, so when you did the CRISPR, you would take out the bad mutations and replace them with a functional gene. And so would that be the knockout, knock-in combo that we're talking about?
Yes. Yep. So the knockout is you take out the function of the gene altogether. That's fairly easy. The knock-in is usually referring to correcting the mutation. So what turns out then is that before the CRISPR, the patient has a single point mutation. After the CRISPR with the donor DNA, if the donor DNA gets integrated correctly, it will cut out the piece that has the mutation. It'll insert the new donor piece that doesn't have the mutation. And so you've knocked in the correction. Alternatively, you could knock in a mutation into a healthy cell line to say, oh, what would happen if this cell line or if this model organism had the mutation?
That makes so much sense. Thank you for clarifying that. Now, there are lots of medical applications for CRISPR technology then. I'm seeing like that's one of them. That's a perfect one. Are there any others that you can think of for treating human disease?
Yeah, this is where things get interesting. And the idea of gene therapies really come into play. So there's several ways to edit the genome in order to treat the patient's genetic mutation. So let's just sort of walk through three possible ways. So in the first scenario, you can correct the mutation that the patient has by including the donor sequence that has the correction in it together with this CRISPR-Cas9 system. This mechanism then, in theory, corrects the patient's mutation. So the second treatment option is to say, okay, well, this patient has a mutation and it results in an abnormally functioning gene. But let's say there's another gene that the body makes that can compensate for that patient's mutated gene, different gene altogether. So what you can actually do is you can turn on a different gene that can produce another protein which compensates for the patient. So let me give you an example here. So the way you would do that is there are genes that turn genes on, and there are genes that turn genes off. So if you target the gene that turns off a second gene, if you delete that gene, that is the repressor, that results in the target gene that you care about increasing its expression. So the net effect of this then is an increase in the expression of the compensated gene. And this is relevant because this is the basis for the first ever approved CRISPR technology. In fact, today, the FDA approved in the United States the first ever CRISPR technology for sickle cell disease. And about 2 weeks ago, it was approved in the UK. So it's just monumental. Wow. And sort of the thought processes here is that the gene that represses fetal hemoglobin, is targeted. And that results in cells increasing the amount of fetal hemoglobin. So they produce fetal hemoglobin. And all you need is for the fetal hemoglobin, which should be turned off by the time we, are born. If you can get about 20% of all the hemoglobin to be fetal hemoglobin, you can eliminate the sickle cell crises and the need for blood transfusions in patients with sickle cell disease. So this CRISPR technology is not changing the mutation that causes sickle cell. It is activating a second gene. Fetal hemoglobin works just as well as regular hemoglobin, and it's able to rescue these patients from the morbidity and the mortality related to these sickle cell crises. So it's just a mind-boggling and really, really cool application for how can you use gene therapy and editing the genome to compensate for a disease. So then, of course, the third mechanism for thinking about how we could treat patients would be just to knock out the mutated gene. So this mechanism is under investigation right now. It's in a trial, a clinical trial, and it's used to treat a disease called amyloidosis. And this is where a mutation in a specific protein causes that protein to misfold. And those misfolded proteins then get deposited in multiple organs, including the heart. And then you get the disease, which is amyloidosis or cardiac amyloidosis. So the goal of this gene therapy is to target that gene and knock it out. And by knocking it out, you reduce the expression of these abnormal deposits, and then you can treat the disease. And the idea and the hope here is that this does seem to work in the preliminary data, but if it does work, then you could treat other diseases of misfolding, and that includes dementia and Alzheimer's. Some forms of Alzheimer's dementia are thought to be misfolded proteins that end up in the brain.
It seems like we're using any pathway we can possibly do. So I like your where we're focusing on the promoter region. I like that for the sickle cell anemia. That's very smart. Because we know I'm a pathologist, so we know that the lab test there's an increased fetal hemoglobin in those patients, and that's just a natural thing that occurs because they need to be able to survive. But we need to bump up that fetal hemoglobin with a promoter region, and so that's really smart. And when you were talking that, it made me think like, well, why don't we just replace the hemoglobin A1 gene? Like why don't we promote that gene? Or and I don't know. I mean, maybe you can talk to some like ethical reason for that, or some. Sorry, I'm going a little off script right now, but
Yeah, no. I think it's more technical, and that is, remember that base editing, that patient correction is a very low frequency event, whereas just having the DNA cut and inactivate a gene is a much easier pathway.
that makes so much sense.
So I was actually thinking about that, and I'm glad you used the term mind-boggling, because it really is. I remember when I did IVF and I saw my embryos at 132 cells under the electron microscope and thinking that was fascinating, and that's like the highest technology. And you're actually talking about assessing these gene sequences within each cell. I mean, it's mind-blowing to me that this is even possible. So how do you actually, like, getting back to kind of the technical aspect, how do you introduce like the CRISPR system into these specific cells or organisms? And are there like certain barriers to the CRISPR delivery to these affected organs?
Yeah, this really is the biggest challenge to the widespread use of gene therapies. That is, how exactly do you deliver the gene, or in the case we're talking about here, the CRISPR machinery. So there's really two major ways of accomplishing this. One of them includes viral vectors, and the other includes nanoparticles, and they're typically lipid nanoparticles. So let's break down each of those. So the viral vector method involves packing the guide RNA, the Cas9, and the donor DNA into a non-replicating version of a virus called adeno-associated virus, or AAV. And in some cases, we would use a non-replicating adenovirus. So the AAV can only take a small amount of DNA package. A much bigger gene or a much bigger machinery can be included in the adenovirus. And so that's sort of how and why somebody would choose the AAV over the adenovirus. These are both non-replicating, so you're not going to give the patient adenoviral disease. But there are some complications we'll talk about in just a second. So usually in these situations, the viral vector with its package of DNA inside the CRISPR machinery is infused intravenously, but there's a predilection for the liver and the heart. So that's just because these viruses tend to affect, infect, I should say, the liver and the heart. So a major side effect of the viral vector delivery system is an immune response to the virus, to the vector itself. And in some cases, that can be lethal. And in the early days of gene therapy, there was a patient who died after receiving an adenoviral gene-based therapy, who was a young college student, and that put a hold on gene therapies for 10, 15 years. So now we're back using these viral vectors in many clinical trials, and there have been deaths associated with them as well. So, what it ultimately turns out to be the frequency of this overwhelming immune response and death still needs to be sorted out. Currently, all of these gene therapies are targeted toward, lethal or near lethal diseases. And so the risk-benefit ratio is in favor of having a potentially lethal outcome as a side effect if you know that the patient's trajectory is going to be death, whether it's in the first 2 years of life or in the first 20 years of life. So kind of to get around this issue of the size that can be packaged into the viral vectors and the immune response, investigators have devised these small particles called nanoparticles that are lipid-based, and these are infused into the blood intravenously, and they're readily taken up by the liver because the liver thinks it's a lipid particle and it needs to sort of take it up and metabolize it. So these are really good for targeting disorders of liver metabolism. If we think about a different class of disorder, blood disorders like sickle cell anemia, but blood cancers are the same general concept, this allows a different mechanism for getting the virus into the cell of interest. Because in this case, you can do a bone marrow aspirate. You can take out the patient's own bone marrow cells, you can harvest them, and then in the test tube, you can treat them with a viral vector so there's no immune response. The other thing is you can enrich for your gene-edited cells, finding those clones where you've corrected the mutation. And then these CRISPR-altered bone marrow cells then get reinfused into the patient, where they then take up residence in the bone marrow and they produce the normal desired gene product. In these situations, though, the patient has to undergo bone marrow ablation before infusing the treated cells. That's like a bone marrow transplant, right? So in this case, they receive agents that suppress and kill the bone marrow, so they can't make white blood cells or red blood cells. And so there's a period of time where they're very immune suppressed and at risk of disease. And then once the bone marrow cells that have been infused take residence and start producing those cells, then their hemoglobin comes up, then their white blood cells come up and things like that. So blood disorders are really a unique and I guess you could say easier disease to treat because you can take the cells out of the body to treat them. Sort of similarly, disorders of the retina are a little bit easier to treat with gene therapy and CRISPR therapies because you can inject the materials into the, just behind the retina. So that's an enclosed space. So you don't have the CRISPR machinery sort of entering all the cells of the body. So these are ways you can direct your therapy to the right cell, at the right time. If you want to target the heart, if you want to target the nervous system or the liver, you need to get your vector into those cells, but not into the other cells of the body, which is always a challenge. And so, fast-paced, most of these technologies are working in the blood space or in limited spaces where you can inject, like the eye, or intrathecal into the spinal cord for, like spinal muscular atrophy or SMA.
That's really awesome. My mind's going crazy with all these connections here, and where we can inject. Now, I'm also wondering, like with the eye, since it's kind of an immunologically compromised space, or not compromised, but it's an immunologically different space than most of the body. Like, is that one of the reasons why you can put the CRISPR machinery in there, or is that, okay, that's the reason.
Yeah. It's more such a localized space. And believe it or not, the ophthalmologist can inject behind the retina, right? Like, it sounds crazy, but they do that. And it's semi-routine. But then all of these, machineries to edit the genome get localized in one space, and they're really only altering the retina and some other cells in that region, versus giving it IV, where it goes to every cell in the body, could potentially pick it up. Earlier,
you'd mentioned a PAM sequence, and I wasn't entirely familiar with the PAM sequence. I figured we'd get into it because you mentioned it. But that serves as the anchor for the CRISPR machinery. And that's what I get out of it. Now, does every gene have a PAM sequence? And are there genes that just simply cannot be targeted?
Yeah, so this PAM recognition sequences, they're just sort of scattered across the genome. They're part of our genome and all organisms' genome. And that's what the bacteria take advantage of. The fact that these PAM sequences exist, the bacteria take advantage of that to design, their RNA CRISPR-like molecules, which then go and target the virus genome and cut it and inactivate the virus. So These PAM sequences have specific nucleotide sequence, and they're just sort of scattered across the genome. But not every region, so every gene has PAM sites, but not every region of the gene has a PAM site close enough to the area that you might want to target. So that's where there could be some areas of the genome that would not be targetable because you can't get the CRISPR close enough to it. So to overcome this issue, researchers have designed CRISPR systems, meaning they've introduced some mutations into the CRISPR system that allow recognition of something that would be a partial PAM sequence, but not the full PAM sequence. And again, that's the anchoring point for the machinery, but the real business end of the machinery is the RNA complementary to the DNA. So even if you make it easier for, you don't need 100% identity at the PAM sequence, let's say you can get it to recognize areas that might have 60% homology, 50%, I'm kind of making up these numbers because I don't know for sure, but something in that range, it can still bind there and anchor. And then the other side of the binding is a very, very tight and specific RNA side, which binds to the DNA. And so that's one way of sort of thinking about how you can engineer the machinery to target more sites. And then there's also engineering that's being made to allow the CRISPR machinery to reach further away. So right now, the PAM sequence has to be within X number of base pairs to the site you want to cut, because it's got to be close enough to anchor the PAM and direct the Cas9 to the RNA binding site. But there are some systems that allow a longer reach in a sense. And again, that allows more sites to be targeted, because the future goal is, if we want to make this into a therapy, which is, going to affect, millions of people, then, we need to make sure that we can target those individuals based on where their mutation resides.
Are there other diseases that that they're currently working on? And you mentioned that it's more readily uptaken by the liver because it's lipid-based. Does that mean that liver abnormalities such as hepatitis C and things like that are more likely to be eradicated? Are they targeting liver things, or what do you see really on the horizon?
Yeah, sure. So sort of in thinking about the vector and the delivery systems and advances there, the liver is a great option to treat because, again, it recognizes lipids in the blood and it can pull them out. And so for diseases that affect the liver, that's a great option. And there's lots of hereditary diseases and metabolic disorders that impact the liver function that could be treated with that methodology. Some of these lipid particles are also under consideration for treatment of heart disorders. If you can inject these into the coronary artery, for instance, or into the coronary sinus of the venous system of the heart, then these, that's sort of trying to get it closer to the organ that you care about if you're targeting, a cardiac disorder. And then these lipid particles are sort of taken up into the blood, into the cells of the heart with the CRISPR machinery and things like that. And so while the lipid nanoparticles are especially helpful for the liver because of its uptake, they can be used in other settings. They can also be injected into the spinal fluid, for instance. And then you could get, these CRISPR machineries into the neuronal cells, for instance, to treat neurological disorders.
And I know, Scott, you mentioned earlier some of the ethics. I was just, as you were speaking, I was thinking how this could really, the train could get out of the station and you could really modify a lot of gene sequences. Do you want to talk a little bit about the ethics? And I know there's been some recent cases in the news about this as well. Do you want to kind of talk about that?
Yeah. So I think the highest profile case of the use of CRISPR technology in a human embryo came out of China a few years ago. something on the order of 5 years ago. I'm not sure exactly on the timing of that. But a Chinese researcher genome edited a fertilized embryo with the goal of introducing mutations that would make it more difficult for HIV to infect the cell. And the mother who was going to carry the child was HIV positive. And so this researcher genome edited a fertilized embryo and introduced this technology into the embryo, made the edits, and then implanted that embryo into the mother who was HIV positive. And that cell then split early on, and the mother had twins, both of them identical, carrying this new mutation that they didn't originally have that would protect them from HIV. So it sounded good. It sounded like a noble goal. But the issue is that in the modern era, with antiviral therapies and with a low transmission of HIV from the mom to the fetus, this was felt to be a step too far because it was editing the genome of a person without understanding what could be the off-target effects. So we've talked earlier about how precise CRISPR is because of its, specific binding to a specific region of the DNA, and off-target effects are low, but especially given the technology that was used 5 years ago or so to create this genome-edited embryo, there could be introduced mutations somewhere in the genome that we have no idea about, that these two children then will pass on forever to their family because we've edited their genome. And so there was a huge outcry. That individual was jailed and in China and ultimately recently got out. But it speaks to the idea that this is doable. Like, you can edit a human genome. You can edit a fertilized embryo. And then the fear is that you're going to edit it to give that individual a certain feature, which might be, some other feature, either intelligence or athletic ability, or, those are harder because there's not a single gene that controls those. But that's sort of the idea, is that you could, they call it a designer baby. I think that's probably an exaggeration. But there is the fear that, you could use these technologies to, change the human race in some way by creating individuals with, unique capabilities.
What are some of the modifications to the CRISPR system or engineering of the system that might actually improve the efficiency of genome editing for human therapies?
Yeah, so there's really been some clever modifications or introduced mutations within the CRISPR machinery that, like, as we talked about before, with the recognizing a portion of the PAM site or being able to extend further, there's other alterations that can be used and that have been introduced recently. And what's so exciting about this is the field is just evolving so quickly and advancing so quickly. Like, every month there's a new idea, a new concept, a new technology. So making everything better and safer. So I think that's really, really cool. And so a couple of these have really helped in the efficiency of the CRISPR process. So one option is to substitute the Cas9 endonuclease for another enzyme. So remember, the Cas9 will make a double-stranded break, and then the cell's intrinsic ability is, hey, I've got to fix this. And invariably, in trying to fix it, it screws it up and you get a loss of function. And that introduced donor DNA is a really, really low frequency event. So if you substitute the Cas9 endonuclease for another enzyme, this is called a nickase, the nickase will actually cut just a single strand, not the double strand, a single strand. So then you don't have a double-stranded break. You don't have this mismatch in trying to repair it. The repair, either it repairs itself or it'll accept the donor DNA to make the repair and introduce that specific mutation at that site. So in the same vein, you can substitute the Cas9 for a different enzyme called a deaminase. And the deaminase doesn't even really cut the DNA. What it does is it switches out a single base, and it'll switch one base to another, and that's going to change the codon, the 3 nucleotide codon that changes then the amino acid. So these deaminases are extraordinarily efficient. And it seems like this may be the future, using either the nickase or the deaminase. These processes are called base editing, and these really offer the promise of improved specificity and efficiency.
Now, you're talking about base editing, and I'm thinking here, like, this sounds really similar to like epigenetics, and especially with the deaminases, because that's part of it. So what kind of similarities are there with that?
Yeah, yep. So for the audience, epigenetics are sort of proteins that wrap around the DNA itself, or that modify the DNA. So DNA is coiled up, it's really tight. If you were to unravel it, it'd be, like longer than whatever, I can't remember the details of that, but super, super long. So DNA is super coiled and compact. And if you want to turn a gene on, the factors called transcription factors that bind to the promoter region of the gene to turn it on need to actually reach the DNA. And if the DNA is all wound up and, it can't reach it. So epigenetics involves changes to the DNA structure that allow either access for transcription factors to get right onto the DNA or inhibit them from getting onto the DNA. And so if you alter the epigenetics, you can make the gene more accessible, meaning more likely to turn on, or more repressed, meaning it'll turn the gene off because the transcription factors can't get there. So that's the idea of, say, epigenetics. And the interesting thing about CRISPR is that you can target some of these epigenetic effects so that you can turn genes on by altering the structure of the DNA, making it easier for transcription factors to bind, or the reverse, making the DNA more compact so the transcription factors can't bind. So this is like just now, like starting to be thought of. And I don't think we've even gone through what are the possibilities for, treatment and therapies, because at that point, you're not cutting DNA, you're altering the structure. Could that be safer? Certainly, you may or may not. Some of these epigenetic changes are actually passed down from generation to generation, but not in the same way as an, autosomal dominant introduced mutation would be. So the idea of altering the epigenetics might be a way that you could impact changes in gene expression to improve a patient's disease without causing, significant off-target effects that could harm them.
As you're talking about that, I'm just thinking like beta thalassemia minor, like you have like the major and minor, and essentially you're amplifying the transcription of that protein, and whereas you can do that, and it's really with the epigenetics, but it's also really cool that even with the CRISPR, I didn't know this, we were targeting epigenetic sequences, but we were also targeting the actual genes. So we're working on both ends of the scope here. It's really, really cool.
Yeah, that's true.
Yeah. So with that, where do you see the future of CRISPR-based gene therapies? Can our medical system even absorb the costs of these therapies? Is this going to be astronomical, or is it going to be okay?
Yeah, so cost is certainly an issue, as these therapies and technologies are very expensive. In some cases, a single treatment can cure the disease. And so biotech companies are keen to recover their profits with a single dose, right? And that's a different paradigm than, hey, you have hypertension, you're going to refill this prescription, every month for the next 10, 15, 20, 30 years of your life, and the company makes the money, right? So if you can treat someone with a one and done situation, then, the companies that design these technologies, the drug or biotech companies, need to make their profit, right? So the costs for some of these gene therapies are in the millions of dollars. And the cost for some of the, curative treatments for other disorders that aren't necessarily gene therapies, those are also in the millions of dollars, again, because it's a kind of a one and done deal. But if CRISPR-based gene therapies are shown to be safe and effective in these rare life-threatening or severe disorders, then I could envision a future whereby gene therapies targeting genes that more broadly are related to the disease or can compensate for the disease rather than the primary disease gene. So if you think about, for example, elevated cholesterol. So there is currently an FDA clinical trial whereby they're using CRISPR technology to base edit a gene, which is in the cholesterol and lipid pathway, which results in markedly reduced cholesterol levels. Even if you don't have a mutation that causes hypercholesterolemia, if you have standard high cholesterol, this therapy would be a treatment for you. And if you can, inactivate that gene, then, potentially with the gene therapy, you wouldn't take any medications because your gene has been permanently turned off and your cholesterol levels are low. Not to the point where you can't make, hormones that require cholesterol, not at that level, but, markedly reduced to the point where you don't need to take a statin, for instance. So, the future goals, and this is certainly the goals of biotech companies and pharma companies, because they don't want to treat the 1 in 50,000 person that's got a rare genetic disorder. They want to treat the 1 in 20 person who's got high blood pressure or high cholesterol or, more, type 2 diabetes, the more common diseases. They want to treat that with a gene therapy. And so that, I think, is where the future will be, with considerations of cost being controllable and with considerations of safety. So really, with that in mind, we'll need to wait and see how the future unfolds. But this future is going to be very exciting and paradigm changing when you think about what medical students are going to be learning 5 years from now or 10 years from now in their pharmacology classes compared to what we learned, with traditional, pharmacology and medications and things like that.
Yeah, thank you so much for your time. It has been such an interesting topic and so cutting edge, and something that is just almost seems impossible to imagine it being successful, but it's so interesting. Thank you so much for your time and for sharing your knowledge with the Ogden Surgical Medical Society.
Well, I'm thrilled to have been a part of the discussion today. Thank you so much for the invitation.
Thank you so much, Marty.
The producers of this show were Clark Madsen, MD and MS, Teresa Puskedra, MPC, and the Ogden Surgical Medical Society Board of Directors. The executive director is Teresa Puskedra. Your hosts were Scott Moore, DO, and Heather Heilson, MD. Editing and sound design were by Owen Service. This is all supported by the Ogden Surgical Medical Society like you. Thank you.